Good morning, everyone. Welcome back, and thank you for joining us. We're glad that you're here. I'm Steve,
And I'm Scott.
and together we are Backyard Musings, the Science and Technology channel.
Yeah, and we're beginning today where we ended yesterday with a health topic, and this one is pretty big. Deals with breast cancer, and this is a cancer that we'd love to see go away. So the more they study about this stuff, the better. So here we go.
Testing thousands of genes in living tumors revealed hidden drivers of aggressive basal-like breast cancer that cell structures largely fail to detect. Inside basal-like breast cancer cells, entire stretches of chromosomes may disappear while others are copied repeatedly. Each altered region can contain hundreds of genes, leaving researchers with a difficult question. Which of those genetic changes actually help the cancer grow? Researchers in Toronto have developed a way to investigate that confusion directly. Would you like to know more?
Using a gene editing system that can both disable genes and increase their activity inside living animals, they identified 81 previously unrecognized genes associated with the growth of basal-like breast cancer, BLBC, one of the most aggressive and difficult forms of the disease to treat.
Yeah, this is wonderful. 81 more threats for cancers.
Yeah,
Not good news.
not good.
Well, finding it is good.
Right.
The findings, published in Nature on July 8th of this year, provide a more detailed view of how abnormal chromosome numbers and rearrangements influence cancer. They could also help researchers identify vulnerabilities that may eventually be explored as treatment targets.
The study was led by Dr. Daniel Schmarmek,
Schramek.
Deputy Director. What is it?
Schramek.
Schramek, Deputy Director of Discovery Research at Sinai Health and Senior Investigator at the Lumenfeld Tannenbaum Research Institute, and Dr. Khalid Al-Zarrani, formerly a postdoctoral fellow at the LTRI and now a faculty member at the Donnelly Center for Cellular and Biomolecular Research at the University of Toronto.
That's some pedigree right there.
Yeah.
BLBC disproportionately affects younger women of color. It is frequently marked by aneuploidy, a condition in which cells contain abnormal numbers of chromosomes or chromosome segments because large sections have been lost or copied multiple times.
Healthy cells depend on carefully balanced amounts of genetic material. When that balance is disrupted, normal cellular functions can break down. Cancer cells, however, can exploit some of these changes to divide more rapidly, survive difficult conditions, and spread through the body, which, that's not good.
The scale of the disruption has made it difficult to identify the genes that matter most. A single duplicated or deleted chromosome region can contain hundreds of genes, and each one may become more active, less active, or disappear entirely. Researchers therefore face the equivalent of searching through a crowded genetic neighborhood without knowing which address contained the cancer driver.
Interesting analogy. The treatment problem is especially urgent because BLBC is also called triple-negative breast cancer. Its tumors lack 3 receptors that clinicians commonly use to classify and target other breast cancers. Without those molecular targets, patients have fewer options for precision therapy.
To sort through the altered chromosome regions, the researchers built on years of work adapting CRISPR, C-R-I-S-P-R, we talked about that, genome editing for the mouse mammary gland, a widely used model for studying breast cancer. CRISPR allows researchers to make controlled changes to genes. In this case, it provided a way to test thousands of candidates inside a living animal and observe which changes encouraged or restrained tumor formation.
The existing platform could knock genes out, meaning it could silence them and simulate what happens when genetic material is lost. Both aneuploidy can also create extra copies of genes, increasing their activity. The system could not could not reproduce that second half of the problem, preventing researchers from fully modeling the chromosome changes seen in BLBC. Dr. Al Zarani, I don't know if we mentioned him earlier or not, but anyway, addressed that limitation during his postdoctoral training with Dr. What was that again?
Schramek.
Schramek, and Dr. Jeff Rana, also a senior investigator at LTRI. He developed CRISPR-Koala, which is a knockout and activation link assay, a system capable of turning some genes off while switching others on within the same mouse. I remember we talked about that CRISPR, you mentioned it earlier, but we talked about that, and I can't remember if that was a story where they were able to pinpoint the treatments rather than just blast in a certain area. They were able to pinpoint.
Yeah.
I think that's
memory. Yeah, pinpointing. Mm-hmm.
Yeah.
That dual function allowed researchers to imitate both deletion and duplication. They could test what happened when individual genes disappeared, as well as what happened when additional copies caused genes to become unusually active.
The researchers used CRISPR-Koala to examine more than 3,700 genes located on chromosomes commonly altered in BLBC. The screening identified 81 genes that had not previously been recognized as cancer drivers. Most would have remained hidden using conventional laboratory methods. 90% of the newly identified genes were not detected in standard cell culture experiments, where cancer cells are grown in dishes outside the body.
The difference show why the living tumor environment mattered. Cancer cells inside a body interact with surrounding tissues, immune cells, blood vessels, nutrients, and changing oxygen levels. Those relationships can influence which genes become important, but many are absent or greatly simplified in cell culture.
One gene, PLGRKT, emerged as an especially powerful driver of BLBC. The researchers found that it helps cancer cells survive in areas deep inside a tumor where oxygen is limited.
Most cells use oxygen to extract energy efficiently from nutrients. When oxygen becomes scarce, cells must rely more heavily on alternative metabolic processes, similar to switching to a backup power source. PLGRKT helped cancer cells make that adjustment, allowing them to continue generating energy under conditions that might otherwise restrict their survival.
This ability did more than keep individual cells alive. The researchers found that it actively supported tumor growth, making PLGRKT a possible candidate for future targeted treatment research. Okay. That one. Interesting.
Yeah.
But yeah, 81 more.
Yeah.
That's not a good number. So,
well, discovering it is the first step to curing it.
Right. But I'm sure that if they've been studying this for a while and it's kind of a step back, right? Finding that many more. But you're right. They gotta find it before they can kill it. Gotta grill it before you can kill it before you can grill it. So
yeah.
Okay.
All right, folks, thanks for joining us. We'll catch you in the afternoon.
Take care, everyone. Thanks.